So as we consider cell signaling, we need to consider the next step in this uh, complicated network of protein interaction, and that is the transduction step. And so again, this is everything now, basically from the receptor to ultimately, in this case, differential gene expression. So there's a lot going on. This is really a very important step, and it's really the heart of how a cell is going to integrate multiple signals from the outside to make quote-unquote a decision on what it needs to do next. Right? This is really the computing power of the cell. And so what's going to happen during transduction? We're going to have a cascades of molecular interactions, right? So this idea that one is going to lead to another, is going to lead to another, is going to lead to another is very, very important. So the multiple steps are an opportunity to amplify the signal, which we'll look at a phosphorylation cascade more specifically to show you how that works. It's going to provide an opportunity for coordination and regulation of the cellular response. So every step, every time one protein touches another, and in some cases multiple proteins converging on a single protein, is an opportunity to coordinate and regulate that response. Do we really want to do this? Or does some other exterior or environmental factor say, maybe we shouldn't do this right now? So at each step, we're going to transduce that signal to a different form, and usually, the majority of the time, that's going to be a protein shape change, right? We're going to change the conformation of a protein to go from inactive to active or active to inactive. And that's going to be our major kind of conduit for information to travel through this process. So if we look at a phosphorylation cascade, a very, very common mechanism to transduce signals, we see the receptor is activated. It's going to activate a relay molecule, which is going to take an inactive protein kinase to its active form. Now remember, a kinase is going to phosphorylate a protein, and that inactive kinase, now active, can activate yet another kinase, making it active. So it then phosphorylates yet a third kinase, and it becomes active, which can then ultimately activate a protein that does something, a cellular response. And so you may say, well, this seems like an awful waste. Why doesn't just the initial protein activate the protein of interest? And what we're going to see is that this actually is an opportunity to amplify the signal. So from a single molecule, we can have hundreds, if not thousands, if not millions of different enzymes or of different proteins activated when we kind of go through this amplification step. But we'll come back to that in a second. But you can see, again, the opportunity, like, for example, in the middle of the screen, you see the RAS protein. So here you have lots of different proteins converging on the RAS protein, and that is a key opportunity then to get input about, do we move forward, do we not? And so when you see an arrowhead, that indicates an activation. When you see a straight line at the end of, like, from RAS-GAP to RAS, that actually indicates inhibition. So you've got two inputs, one that says stop and one that says go. And when the summation occurs, you get one or the other, right? So this is, again, the computer component of kind of the cellular interactome, the network. Now, the other topic that we need to introduce at this point is the idea of the second messenger. So the extracellular signal, the ligand that originally binds the receptor, we call that the first messenger. And we generally don't even talk about the first messenger because we're usually talking about a hormone or whatever generic ligand we're referring to. But second messengers then are going to be an integral component of the transduction pathway. And so these are going to be small non-protein molecules soluble in the cytoplasm that spread or diffuse, right? So they're going to move out through diffusion, and they're going to activate something else based on their concentration change ultimately. So common second messengers, and the examples that we're going to look at, are cyclic AMP and calcium. So cyclic AMP is one of the most widely used second messengers. So adenylyl cyclase is going to be the enzyme that we oftentimes see in the plasma membrane. It's going to convert ATP. It's going to hydrolyze all of those phosphates down to the last one to get cyclic AMP in response to an extracellular signal. So this is what that molecule looks like, right? And we've seen cyclic AMP before when we talked about the activation of the the cap protein in prokaryotes when we looked at the lactose operon. So go back and review that and see cyclic AMP there as an activator of that particular operon. So many signal molecules can trigger the formation of cyclic AMP, and oftentimes these are associated with pathways that have G proteins. All right. So we oftentimes will engage a protein kinase, and then we'll get further regulation of cell metabolism is provided by the G protein system that inhibits adenylyl cyclase. Okay, so what are we looking at here? Let's look at the pictures. The pictures are the best. So we have our first messenger that activates our G protein receptor, which we've already talked about. Our G protein then is going to bind a GTP, becomes active, right? And then one of the proteins that it's going to interact with could be adenylyl cyclase, depending on the pathway. Adenylyl cyclase then is going to take ATP and make cyclic AMP. As the concentration of cyclic AMP increases in the cytoplasm, that second messenger then can interact with protein kinase A that's going to do whatever is downstream on that pathway. That may initiate a phosphorylation cascade or whatever, but then it will ultimately lead to a cellular response. So cyclic AMP is our non-protein, water-soluble second messenger as our first example. Our second example is calcium, okay, and IP3. So calcium ions act as a second messenger, and one of the properties of the ER that I think I mentioned only very briefly, if at all, is that it sequesters calcium. So the lumen of the ER is actually much higher in calcium concentration than the cytoplasm in general. And so what we can do is we can kind of tap into that to release a burst, a wave of calcium that can act as a second messenger. And in fact, this is one of the ways that your muscles work. So there's a special ER in your muscles called sarcoplasmic reticulum, and these bursts of calcium are very important to how your muscles function. So what we can do is we can relay a signal transduction pathway by triggering an increase in calcium in the cytosol. So we can do that, and one of the classic examples is using inositol triphosphate, IP3, and diacylglycerol, DAG, as additional second messengers. So let's take a look at that pathway. So again, starting with our G protein coupled receptor, we can, in this case, initiate activation of phospholipase C. Now, phospholipase C is going to cleave PIP2 into DAG and IP3. IP3 then is released into the cytoplasm. As it increases in concentration, it's more likely then to engage a calcium-gated ion channel in the ER. So we see how all these things come together inside the cell and out. So once that calcium-gated ion channel once that IP3-gated ion channel is activated, calcium is allowed to move down its concentration gradient out of the ER, and we get this wave of calcium in the cytoplasm. And that can activate all kinds of proteins. The major protein that we think about, or that I think about when we talk about calcium pulses, is calmodulin. And that's a classic protein for binding calcium and then activating it to do something else, to activate another protein or ultimately turn on gene expression. So what is that final output? And so in general, this is why I teach gene transcription and translation before I talk about this stuff, because you understand now that there are a number of transcription factors and all kinds of protein elements that are necessary to turn on a gene. So turning on those transcription factors, activating them, or turning on the genes that produce those transcription factors to activate certain genes is oftentimes the output response from these signal transduction pathways. All right? So the synthesis of an enzyme or other proteins, turning on genes or on or off in the nucleus, is kind of like the way, the end result, the output, the response that the cell is giving. Not always, but in many cases. So again, if we go back to our generic reception transduction response, we see reception, receptor binding its ligand, transduction, whatever form it takes there, ultimately leads to the activation of a transcription factor. That transcription factor, now active, can engage the transcription machinery and basically turn on a gene, and that novel gene expression is going to do something in the cell. All right? Other pathways, instead of activating genes, will actually activate enzymes. So again, if you go back to the idea of the control of gene expression, we had many, many levels of the control of gene expression. Only one level is at transcription. So you can produce protein but keep it inactive, and that's one level of quote-unquote expression. And this would then be the end result of turning on those proteins. So for example, if you bind epinephrine, so adrenaline, to the G protein-coupled receptor, you can actually then talk about the mobilization of glycogen to glucose. That this is a time now to move the storage of energy into a utilizable form of energy that we can feed into cellular respiration. Because if I've got adrenaline flowing through my veins, I'm in a fight or flight. I need that energy to do something to keep me alive. And so you can see here that initial activation of the G protein going through these this phosphorylation cascade. You can see literally orders of magnitude increase each step of the way to get more and more molecules recruited to this signal to ultimately get the response of converting glycogen into glucose. All right. So that can be so many different forms. Again, in this case, we've got a G protein-coupled receptor activating whatever protein is expressed in that particular cell. In this case, moving towards formin, activating it so that microfilaments are synthesized and actin subunits are recruited to move a cell in a particular direction. So this goes back to our idea of how does a yeast cell move in a particular direction? Well, localized receptors activate a transduction cascade that changes something, in this case, the cytoskeleton, and hence the cell moves.